Hello everyone, myself Varna, and today I am here to take about fructose and galactose metabolism. So you all know what is glucose, fructose, galactose, then what is glycolysis and gluconeogenesis. All this you are familiar. So today let us see the metabolism upon fructose and galactose. So first, fructose metabolism. So what is fructose? Fructose is a ketohexose sugar which is present in fruits, honey and sucrose. It is also present in soft drinks in the form of corn sugar. Corn sugar has high fructose content, and this fructose is sweeter than sucrose, and this fructose is promptly metabolized by the liver. So, the metabolism is mainly taking place in the liver, and it is taken in a faster rate. It is promptly metabolized by the liver. So, let us move on to the metabolism. So, the main site of this fructose metabolism is liver. Liver is the main site of fructose metabolism. Fructose is phosphorylated to fructose 1-phosphate by the fructokinase in the liver. So, the first enzyme coming into action is fructokinase. This fructokinase is converting fructose to fructose 1-phosphate. By phosphorylation, it is getting converted into fructose 1-phosphate. And then this fructose 1-phosphate is cleaved by the liver aldolase. Next enzyme coming is aldolase. Aldolase B next is acting upon it to form DHAP, dihydroxyacetone phosphate, and D-glyceraldehyde. So, DHAP you are familiar in the glycolysis. D-glyceraldehyde also is an intermediate which is present in glycolysis and gluconeogenesis. This D-glyceraldehyde is phosphorylated by the glyceraldehyde kinase or the triokinase to D-glyceraldehyde 3-phosphate. Let us see. First, this fructose from the diet, it is getting converted into fructose 1-phosphate by the enzyme fructokinase. Fructokinase is making the conversion from D-fructose to fructose 1-phosphate. Then the aldolase B enzyme is converting it into D-glyceraldehyde, and that is getting converted into glycerol and then to glycerol 3-phosphate. And then finally, this glycerol is entering into the synthesis of triacyl glycerol. This D-glyceraldehyde, which is an intermediate, is getting converted into glyceraldehyde 3-phosphate. In other way, this D-glyceraldehyde is also getting converted into glyceraldehyde 3-phosphate, which is an intermediate present during glycolysis, gluconeogenesis, as well as during glycogen metabolism. The enzyme is triokinase enzyme. Triokinase is converting D-glyceraldehyde to glyceraldehyde 3-phosphate. So, glycolysis is synthesis of pyruvate from glucose. Glycolysis, glucose is broken down to pyruvate. Gluconeogenesis is synthesis of glucose from a non-carbohydrate substance like pyruvate. Then glycogen, you all know what is glycogen. Glycogen is a stored form of glucose. So, extra glucose is stored as glycogen, and when there is a shortage of glucose, this glycogen is broken down by glycogenolysis to glucose. Then glycogen synthesis is called as glucogenesis. Synthesis of glycogen from glucose is called as glycogenesis. Glycogen, extra glucose is stored as glycogen, and when there is a shortage of glucose, glycogen is broken down through glycogenolysis to form glucose, lysis of glycogen. So, this glyceraldehyde 3-phosphate is intermediate of all these. So, that is synthesized during the fructose metabolism, glyceraldehyde 3-phosphate. And a disease condition called as fructosuria is due to the deficiency of fructokinase. Whenever there is an enzyme deficiency of fructokinase enzyme, it can lead to fructosuria. And fructose in hereditary fructose intolerance is due to the deficiency of aldolase B enzyme. These two trioses triose phosphate, that is DHAP and glyceraldehyde 3-phosphate, they may enter into the glycolytic pathway, gluconeogenesis or glycogenesis according to the metabolic state of the tissue. So, according to the availability of glucose, these intermediates like DHAP and glyceraldehyde 3-phosphate may enter into the glycolytic or gluconeogenesis or glycogenesis pathway. If glucose is not available, it can enter to gluconeogenesis or glycogenesis. If glucose is there, then it can enter, if the glucose content is plenty in that tissue, it can go to glycolytic pathway, glycogenesis. Next, let us see about the fructokinase enzyme. Fructokinase has high affinity of fructose. Fructokinase phosphorylates the substrate at the first position, where the hexokinase action is on the 6th position. Then fructokinase is not dependent on the insulin or hormone. So, the fructose is rapidly utilized by the normal cells because the fructokinase is independent of insulin. Theoretically, fructose will be better utilized in patients with diabetes mellitus because the first few enzymes of the fructose utilization do not require insulin. So, fructokinase and aldolase B, they do not depends upon insulin. So, those who are insulin deficient, like in the diabetic patients, since these two enzymes are not depending upon the insulin, so this fructose will be better utilized in diabetic patients. Fructose will be better utilized in diabetic patients. Fructose rapidly enters the tissue, leading to enhanced fatty acid synthesis, raised serum triglycerides and increased LDL cholesterol level in the blood. All these are atherogenic and harmful. This fructose, since it is insulin independent, it can rapidly enter into the tissues and leading to the synthesis of fatty acids and triglycerides. So, this in turn will increase the LDL level, which is a bad cholesterol. LDL cholesterol is a bad cholesterol which is leading to heart attack and it is atherogenic. So, these are atherogenic and is harmful. LDL cholesterol is getting deposited onto the walls, heart walls is thickened by the LDL cholesterol. So, that is why it is called as bad cholesterol. So, this fructose can indirectly increase the LDL cholesterol. The utilization of fructose by fructokinase then by aldose bypasses a step of the glucokinase and phosphofructokinase 1, which is activated by insulin. This explains why fructose disappears from blood more rapidly than glucose in diabetic suspects. This is because they are independent of insulin. Apart from glucose, this fructose is getting disappeared from the blood. They are rapidly metabolized than glucose. Why there is an increase in TG during fructose metabolism? Fructose metabolism in liver bypasses the phosphofructose kinase control point, hence fructose increases the flux of glycolytic pathway leading to lipogenesis. Since these it is not insulin dependent, they can be easily utilized and the first two steps is bypassed. Glycerol phosphate required for tag synthesis is provided by the metabolism of fructose, leading to the increase in tag pool in the body. Fructose and heart diseases. High fructose levels are known to be related to aging and myocardial infarction. Fructose participates in glycation 10 times faster than glucose and decreases the antioxidant contribute effect in the ischemic heart diseases. So, this fructose, since it is rapidly utilized because it is independent of insulin and it can be utilized rapidly in a faster rate than that of glucose. It can lead to glycation. Glycation, that is what do you say, glucose is getting incorporated into the RBC cells. RBC cells, when there is more amount of glucose in our blood, glycation occurs. Glucose is incorporated into the cells, that is glycation. So, fructose is participating in this, is increasing this glycation process faster than that of glucose, because it is not depending upon insulin. Insulin is the one which will decrease the glucose, and it also, this fructose also decreases the antioxidants. Antioxidant is that are the substances which helps in the prevention of cell lysis. It prevents the cell from attack of other foreign objects or free radicals. So, it is indirectly, this fructose is causing the increase of LDL cholesterol. It is helping in LDL synthesis, and it is causing heart disease. Next is polyol sorbitol pathway. The sorbitol or the polyol pathway is the formation of fructose from glucose. Fructose formed from glucose, that is sorbitol pathway. Aldose reductase reduces glucose to sorbitol. In liver, ovaries and the sperm cells, these are the second enzyme, sorbitol dehydrogenase, that can oxidize sorbitol to fructose. So, the energy provided to the sperms, ovaries are from fructose. For the sperm cell to move, fructose is the main source of energy. So, for there is a requirement of synthesis of fructose in these cells. So, that is by the sorbitol dehydrogenase helping in this step. The pathway from sorbitol to fructose in the liver provides a mechanism by which dietary sorbitol is converted into fructose. In liver, ovaries and sperm cells, there is a second enzyme called as sorbitol dehydrogenase, which oxidizes the sorbitol to fructose. So, fructose is the main source of these cells, source of energy for these cells. So, that is synthesized from sorbitol by the sorbitol dehydrogenase enzyme. So, this pathway is called as sorbitol pathway. This pathway from sorbitol to fructose in the liver provides a mechanism by which dietary sorbitol is converted into fructose. Glucose is getting converted into sorbitol by the aldose reductase enzyme by utilizing NADPH. Then this sorbitol, with the help of sorbitol dehydrogenase, which is an enzyme which is found in the cells especially in the liver, ovaries and the sperms, that is getting converted into fructose. So, fructose is available for the energy needs for those tissues and cells. Next, let us move on to the disorders associated with fructose metabolism. That is essential fructosuria. Fructosuria, as I said before, it is due to the deficiency of the enzyme fructokinase, the first enzyme. Fructokinase enzyme is deficient. Essential fructosuria is a rare and genetic disorder which is caused by the deficiency of the enzyme fructokinase. In this disorder, the fructose cannot be converted to fructose 1-phosphate. There is no toxic metabolites of fructose accumulate. No toxic metabolites of fructose accumulate in the liver, and the patient remains nearly asymptomatic with the excretion of fructose in urine. So, there is no other toxic metabolites of the fructose which is getting accumulated in the liver, and this patient is asymptomatic. No symptoms are being shown except the excretion of fructose in urine. Next one is hereditary fructose intolerance. Fructose intolerance, it is an autosomal recessive inborn error of the metabolism which is incident of the diseases 1 in 20,000 births. 1 in 70 persons are carrier of these abnormal genes. 1 in 20,000 will be having the disease, 1 in 70 will be the carrier of the gene. Seen when sucrose, which contains fructose, is introduced into the diet, especially in infants, usually around the month of 6. That is the age when which we are introducing solid substance to the infants. Up to 6 months they are fully dependent upon milk. So, when we are introducing a diet to these infants who is having deficiency of aldolase B enzyme, that is fructose intolerance, there will be intolerance of the fructose content. Like fructose is mainly in the form of sucrose. Sucrose is containing both glucose and fructose. It is due to the deficiency of enzyme aldolase B. Fructose 1-phosphate cannot be converted into DHAP or glyceraldehyde. Aldolase B is deficient in fructose intolerance. Hereditary fructose intolerance is an autosomal recessive inborn error which is seen in 1 in 20,000 births, and 1 in 70 will be the carrier of these genes, and it is mostly shown when a diet is given to the infants, especially around the age of 6 months. Those who are having fructose intolerance, they will be lacking the enzyme aldolase B, and this aldolase B with its deficiency we cannot convert. Fructose 1-phosphate is not getting converted into DHAP and glyceraldehyde. So, there is accumulation of fructose 1-phosphate in the blood. This will result in inhibition of fructokinase as well and impaired clearance of fructose from the blood. Fructose will not be utilized, there is intolerance, excessive fructose will be accumulating in the gut. So, this results in the inhibition of fructose kinase and an impaired clearance of fructose from the blood. The clinical findings of hereditary fructose intolerance include accumulation of fructose 1-phosphate leads to liver and kidney damage, hypoglycemia due to inhibition of glycogenolysis, breakdown of glycogen, and gluconeogenesis. Then vomiting and loss of appetite will be seen. The infants often fail to thrive. There will not be growth and developments in infants. Then hepatomegaly and jaundice may occur. Then fructose is also excreted in urine, which gives positive Benedict's test. Benedict's test is a reducing test which is done in order to see the presence of reducing sugars like glucose, fructose, etc. So, that will be positive because fructose will be excreted through urine. Then treatment is elimination of fructose from the diet. So, completely eliminate the foods which contains fructose. The effect of hyperglycemia on sorbitol metabolism. Next, let us see the effect, how hyperglycemia is affecting the sorbitol metabolism. Retinal lens and nerve tissues are not insulin dependent. Large amount of glucose may enter these cells during hyperglycemia. During hyperglycemia, large amount of glucose is getting inside the retina, lens and the nerve tissues, since they are not insulin dependent. For example, hyperglycemia during diabetes. Hyperglycemia is seen in diabetic patients. So, they will be having high glucose value in their blood. Normal value 70, 110 up to 140. So, these diabetic patients will be having greater glucose value greater than 140. So, this larger amount will be entering in retinal lens and nerve tissues. So, there is increased glucose as well as this glucose is getting converted to sorbitol, and this sorbitol, with the help of sorbitol dehydrogenase, is getting converted back to fructose. So, increased glucose means increased sorbitol will also be there. This causes osmotic damage leading to cataract formation, peripheral neuropathy, retinopathy and nephropathy. So, increased sorbitol means more amount of water retention, osmotic pressure will increase. So, all these osmotic damage will be seen, which will lead to cataract, then nephropathy, neuropathy and retinopathy. Sorbitol increased sorbitol amount means there will be more amount of water will be coming inside that cell, which would causes osmotic damage leading to cataract, nephropathy, neuropathy and retinopathy. Drugs inhibiting aldose Drugs inhibiting aldose reductase improve the peripheral nerve functions in diabetes. So, there are certain drugs which will inhibit aldose reductase enzyme and it can improve the nerve functions in diabetes. But this cataract and all is much complicated. Here you can see during if the glucose hyperglycemia, more amount of glucose will be there in the blood. In blood the glucose value is elevated, hyperglycemia, then more amount of glucose is entering into the nerve, lens and the kidney tissues. So, more amount of glucose is entering means more amount of sorbitol. Glucose is getting converted into sorbitol, which is aldose by the enzyme aldose reductase enzyme. So, more amount of glucose means more amount of sorbitol, and more amount of sorbitol it will attract water. More amount of water is also getting inside these tissues, which will increase the osmotic pressure and osmotic damage is occurring in those tissues. So, lens and nerves and the kidney cells are damaged. Sorbitol accumulates in these cells causing strong osmotic effect. Then there will be cell swelling. More amount of water means the cell will swell as a result of water retention. So, it can damage those cells. So, there are certain drugs which can reduce the aldose reductase activity and therefore revert the process of conversion of glucose to sorbitol and can protect these cells. So, this is with the sorbitol. So, increased glucose means, that is hyperglycemia means increased amount of sorbitol which is mainly seen in lens, nerve and kidney tissues. So, all these are dependent are having sorbitol dehydrogenase, and this sorbitol accumulation is leading to accumulation of water as well. Water retention is there and causing cell swelling, which is causing osmotic damage to those cells. Same thing in the testis also. In the testis, the seminal vesicles, which is fructose is converted into glucose through sorbitol formation by aldolase reductase and sorbitol dehydrogenase. Free fructose is seen in large quantities in seminal plasma. The energy of mobility of spermatozoa is mainly derived from fructose. And this fructose is secreted by the seminal vesicles. Azoosperma may be also blocked in the duct. In such patients, fructose is estimated, and if it is present, the block is above the seminal vesicular duct. The main source of energy for the sperms to move is from the fructose. The energy of mobility of the spermatozoa is mainly derived from fructose, and this fructose is secreted by the seminal vesicles, and the fructose is converted into glucose through sorbitol formation by aldolase reductase and sorbitol dehydrogenase enzymes. So, fructose is secreted by the seminal vesicles. In such patients, fructose is estimated, and if it is present in the blockage and the blockage is above the seminal vesicle duct. If fructose is present and if it is present, the block is above the seminal vesicles, and if fructose is absent, the block is after the seminal vesicles. Deficiency of fructose correlates with the male infertility. So, male infertility may be due to deficiency of fructose correlations. In the seminal vesicles, glucose is entering and is getting this glucose entering from the blood to the seminal vesicle. It is getting converted into sorbitol by the aldolase reductase enzyme and then to fructose by the sorbitol dehydrogenase enzyme, sorbitol pathway, because fructose is the main source of energy for the spermatozoas to move. Sperms are moving with the help of fructose. So, fructose need to be synthesized in the seminal vesicles. It is with the help of the enzymes like aldolase reductase and sorbitol dehydrogenase. From glucose, it is getting converted to sorbitol and then to fructose. Glucose from the blood, it is entering to the seminal vesicles, and that is converted into sorbitol, and sorbitol to the fructose. Fructose is the main energy for the spermatozoa for its movement. So, this is just a summary of fructose metabolism. We will just revise what we said, that is fructose getting converted into fructose 1-phosphate with the enzyme fructokinase, and the fructose kinase, if it is deficient, then it is called as essential fructoseuria, that is lack of fructose kinase, it is autosomal recessive, then fructose is getting accumulated in the urine. Next is for fructose 1-phosphate is getting converted into glyceraldehyde by the enzyme aldolase B. Aldolase B enzyme is getting converted, converting fructose 1-phosphate to glyceraldehyde. Glyceraldehyde is then getting converted into glycerol and finally to glycerol phosphate, and it is entering into the synthesis of triazyl glycerol or phosphoglycerides etc. For the synthesis of all those compounds it is entering. When there is a deficiency of aldolase B, then the condition is called as hereditary fructose intolerance. It is an autosomal recessive, 1 in 20,000 births. It is due to the deficiency of aldolase B, leading to intracellular trapping up of fructose 1-phosphate is not getting converted into glyceraldehyde. So, fructose 1-phosphate is getting accumulated, which in turn inhibits the action of fructose kinase enzyme. Causes it causes hypoglycemia, vomiting, jaundice, hemorrhage, hepatomegaly, renal dysfunction, hyperuricemia, lactic acidemia, or all those conditions. Then fructose, sucrose and sorbitol can cause hepatic failure and death, because sorbitol is getting accumulated, more of water retention, more of cellular damage due to osmotic pressure, more of water is getting inside due to the conversion, due to the accumulation of sorbitol. So, it is damaging the nerves, kidney cells and also the lens, nephropathy, neuropathy, retinopathy, all these are caused. Then this glyceraldehyde is entering into the glycolytic pathway or gluconeogenetic pathway or glycogen metabolism. It is an intermediate of all these three. Depending upon the status of the tissue, it can enter either to the gluconeogenesis, glycogen metabolism or glycolysis.